Okay, I think we have 1 minute past, and I think a lot of people have already joined, so I'll not take a long time to study. So hello, everyone. Warm welcome to the Biologically Speaking webinar series. I'm Shaunak, a postdoctoral fellow at the National Cancer Institute, NIH, in the United States, and a co-founder of this platform, Biologically Speaking, which we formed in mid-2020 during the pandemic with a group of PhD students and postdoctoral fellows. So we host scientific talks in the form of webinar, and all of our previous talks are archived on our YouTube channel, where we live stream our session as well. And if you'd also like to be a part of our initiative, please write to us via our website at www.biologicallyspeaking.com, or you can email us to our ID as well. So we at Biologically Speaking has initiated a new series, CROSSTALK, partnering with Dr. Mohit Jolly from the Indian Institute of Science, Bangalore. And together we discuss behind the scenes of collaborations where we talk to scientists and their trainees who got benefited from the collaboration, and how to initiate and sustain a productive collaboration in the scientific field. Today is the third session of CROSSTALK, and I'm absolutely delighted to have our panelists who are distinguished scientists in the field and have been collaborating for a long period of time. And before I introduce our panelists, a couple of housekeeping things to remind. Please keep your microphone muted and videos turned off during the session. And since this is more of a discussion, we encourage our audience to engage and interact, and you can post your questions for our speakers using the chat box, and I will moderate them at the end of today's session. With that, our first panelist for today's session is Professor Kavita Babu, who is currently a Wellcome Trust DBT Senior Fellow and an Associate Professor at the Center for Neuroscience at the Indian Institute of Science, Bangalore. Kavita performed her doctoral studies from the Institute of Molecular and Cell Biology in Singapore, King's College London, and the Temasek Life Science Laboratories. And after a PhD, Kavita went on to do her postdoctoral research at Professor Josh Kaplan's laboratory at the Massachusetts General Hospital in Boston. And in August 2011, Kavita joined IISc and Wellcome Trust DBT India Alliance Intermediate Fellow, and was also a recipient of the DBT Innovative Young Biotechnologist Award. After spending more than 7 years in Mohali, Kavita moved to IISc, and their lab is focused in understanding the C. elegans nervous system and regeneration. Our next panelist, Professor Anindya Ghosh-Roy, who's currently a Wellcome Trust DBT India Alliance Senior Fellow and a professor at the National Brain Research Center at Haryana. Anindya earned his PhD from the Tata Institute of Fundamental Research, followed by a postdoctoral training at the Columbia University and at UC San Diego, followed which he moved back to India to set up his independent laboratory at NBRC, Haryana in 2013, where his research group is focused on understanding neuronal development and regeneration using C. elegans. Professor Babu and Professor Ghosh-Roy both worked with C. elegans and has been collaborating for a long period of time and published a lot of papers together. And to discuss some of their prolific work, we have trainees who have been trained in their respective labs and were instrumental in driving their studies. And we'll be discussing their journey today in today's panel. For that, we are joined by Dr. Pratima Pandey, a PhD from the Institute of Microbial Technology, Chandigarh, and was also a postdoctoral research associate in Professor Babu's lab at IISc when they have done the work that they will be sharing today. And Pratima is currently a DST-funded research scientist at IISc currently. Our next panelist is Dr. Anuradha Singh, who completed a master's in molecular and human genetics from Bangalore University, and also pursued a PhD in Dr. Kavita Babu's lab at IISc and was a part of this collaborative effort. Dr. Singh is currently working at the Patents Division of CSIR in India. And last but not the least, we have Harjot Kaur, an electrical engineer as an undergraduate and currently working on dendrite regeneration in Professor Ghosh-Roy's lab at NBRC, Haryana. With that, thank you everyone for taking the time to be a part of our initiative, and a warm welcome to all of you to Biologically Speaking, and to all our audience for tuning in today. I would now invite, I think Harjot, you will start presenting, I guess. And to all the trainees who, Harjot, Anuradha, and Pratima, who will be introducing their work and we'll have our discussion after the talk. And to our audience, this is your chance to ask questions relevant to their work and also about their collaborative initiative. With that, over to you.
Actually, I'll start.
Okay, sure, go ahead.
Yeah. So you can, so my screen is visible?
Yes.
Okay. So good evening all. Thanks to the organizers. And so today we are going to discuss our work which was a collaborative work with Dr. Anindya Ghosh-Roy's lab. And we here deduced the role for dopamine autoreceptor DOP2 in C. elegans locomotion during ethanol stress. So dopamine, we all know, it is a feel-good neurotransmitter. I think it is a household name now because everyone knows what dopamine does. And when we are under any kind of sedation, there is increase in the levels of dopamine. If we are depressed, there is decrease in the levels of dopamine. So along with that, it is also involved in various other functions, such as movement, in defense, and learning and memory. So along with this, if we look at the effects of alcohol, it also affects movement. It also gives us like our speech is impacted and memory will be gone after like a long intake of alcohol. And even in C. elegans, we find that there is in defect in movement. So we'll focus on movement here. And actually, these kind of behaviors or effects of dopamine and alcohol have been found. However, the circuit underlying these are not very clear. So coming to C. elegans, it is a very good organism to for such kind of studies, since here the neurons are just a handful of neurons, and this dopaminergic system, that is even more concise, just 8 pairs of neurons, whereas in mammals there are almost like 5 lakh dopaminergic neurons. So this is a very good organism to study the circuits. And coming to the dopamine signaling in C. elegans, so the molecules for dopamine signaling in C. elegans, they have been deduced for quite long time, since here most of the sequencing, it was finished, and it was the first organism to be sequenced around like 25 years back. So that was great thing about C. elegans. And here, various molecules, they were studied for their role in different kind of behaviors. And DOP2, it was also found to be participating in associative learning and in anterior touch habituation. And however, still the underlying circuitry was not known. So I have been working with DOP2 before also, like before joining Dr. Kavita's lab. And so I was still interested in DOP2, and Kavita gave me a heads up that, okay, you can work on this. And so now I wanted some kind of behavior or some kind of assay which we can visualize very easily, and we can deduce the underlying circuit for this DOP2 signaling in C. elegans. And so hence, since dopamine is associated with locomotion, and it is associated with alcohol, and it is also associated with feeding in C. elegans. So we actually brought together all those 3 behaviors, and we devised an assay where we exposed the worms to very high levels of ethanol, and the plates were kind of dry as compared to normal assay plates, and the worms were exposed to like they were starved during this time. So we performed assays for 120 minutes, and we didn't find anything very substantial. So the assays were working, but the behavior wasn't very distinct. So one fine day, what happened that I left the lab after performing the assays, and during this time, actually, we had this kind of thing in the lab that people who left the plates outside after the working hours, so they were fined, since we were having lots of contamination problem in the lab. So at night, when I reached home, I realized that I have left the plates outside. And so next day, I made it sure that I went in very early in the morning so that I can discard my plates myself, and no one else figures out that. So when I went to the lab and I looked at my plates, it was very amazing and surprising since our dope-2 plates, so they were having these kinds of concentric circles, whereas wild-type worms, they were having these sinusoidal kind of tracks, and the worms were, in case of wild-type, the worms were not there. And in case of this mutant, the worms, most of the worms, actually we start with 10 worms, and most of the worms were still on the plate. So that was kind of our eureka movement. And I just went to Kavita and I told her that this is what we have found. And it was consistently on all the triplicate plates that we had used for our essays. And then Kavita was also quite excited. And meanwhile, during that time only, Anuradha had joined our lab as a graduate student. So then Kavita asked me to take her help. And then she started working with me. And together, then we deduced the underlying circuit, and then we collaborated with Dr. Anindya's lab also. So now Anuradha will take over from here. So Anuradha.
Just a minute, I'll share my screen.
Okay.
Okay. Thank you, Pratima, for beautifully introducing the ethanol-induced sedative behavior. That was specifically dependent on dope. That was specifically seen in the dope-2 mutant animals. We did a series of rescue and mutant analysis experiments, and we validated this theory that indeed this behavior was dope-2 dependent. We again did a series of experiments, like we exposed the worms to high levels of exogenous dopamine, and through FAP analysis, we validated that the behavior that we were observing is due to the increase in the levels of dopamine. Next, we wanted to see which predicted circuitry is dope-2 functioning through, and we wanted to narrow down to the neuron that through which dope-2 functions to control the EIS behavior. And again, through our series of rescue and mutant analysis experiments, we narrowed it down to the PDE neuron, which is already introduced by Pratima in her previous slides, that it is the only posterior dopaminergic neuron present in C. elegans. And we hypothesized that dope-2 might be functioning through PDE, leading to increase, playing a regulatory role, which leads to increase, in its absence, leads to increase in the levels of synaptic dopamine. Okay. Now, as I've already shown you that dope-2 might be functioning, we were hypothesizing that dope-2 might be functioning through PDE neuron. On narrowing down to this circuitry, what we found is that PDE forms very strong unidirectional synaptic connections to a interneuron, that is DVA. DVA is situated postsynaptic to PDE in the most posterior region, that is the tail of the worm, and it is a single neuron that has been previously shown to both positively and negatively regulate the locomotion of the worm. So next, as I've already told you that we were narrowing down our circuitry to the PD, that dope-2 might be functioning through the PDE sensory neuron, and dope-2 plays a autoregulatory role in regulating the levels of dopamine at the synapse. This dopamine goes on and binds to its receptors present on the DVA interneuron and excites it, leading to the activation of and secretion of a neuropeptide, NLP12. This neuropeptide goes on and binds to its receptor present on the cholinergic motor neuron, regulating the release of neurotransmitter acetylcholine at the neuromuscular junction, in turn regulating the hyperactivation of the muscles and leading to regulation of the locomotion of the worm. So we wanted to devise an experiment to rule out that PDE neuron is the only neuron through which dope-2 is functioning. And first we thought of doing the electrophysiological studies, and the lab we collaborated with had some issues in performing this behavioral experiment, as it is a complex kind of experiment, and they were not able to see the behavior. So we had to drop down that idea, and the next experiment, and I think it was kind of our last resort, but we decided to ablate. We decided if somehow we can ablate the PDE neuron, we we can validate our hypothesis that yes, DOPE2 is functioning through PDE to regulate this ethanol-induced sedative behavior. So we were fortunate enough at that time that we attended this Indian Worm Meet where Harjot was presenting her lab work, and we got exposed to the line of work that was done in their lab, and how it could significantly lead us to proceed with our experimental design. And thus here where the idea of collaboration kind of originated, and both Anindya and Harjot, they warm-heartedly welcomed us to their lab, and both of them have guided me and provided their support during this whole experimental procedure. And now coming back to my hypothesis, coming back to my hypothesis, what we were proposing is that if somehow we could ablate PDE neuron, then the ethanol sensitivity should no longer be present. And indeed, when we ablated PDE neuron in DOPE2 mutant background, we saw that the worm significantly rescued from the EIS behavior, while the mock ablated worm were still showing the DOPE2 mutant behavior. Now coming to the model of my whole study, what I have shown you that DOPE2 is functioning through PDE neuron to regulate the downstream circuitry. If DOPE2 autoreceptor is present, it has an autoregulatory role and functions in a negative feedback loop to regulate the release of dopamine at the synapse, and thus maintains the normal functioning of the downstream circuitry. What happens when what happens in DOPE2 mutants is that in the absence of DOPE2 autoreceptor, there is enhanced release of dopamine, unchecked release of dopamine at the synapse, and which leads to overexcitation of the downstream circuitry. So DOPE2 goes on and binds to dopamine receptors present on the DVA interneuron, which in turn leads to its overexcitation and activation of a neuropeptide NLP12, and leads to its enhanced release. This neuropeptide goes on and binds to its receptor CKR2 that is present on the cholinergic motor neuron, which in turn regulates, or I can say enhances, the release of acetylcholine at the neuromuscular junction. This acetylcholine leads in increased release of acetylcholine at the neuromuscular junction, leads to muscle overexcitation and paralysis, further leading to the EIS behavior that we observe in DOPE2 mutant animals. Now I would like to hand over the presentation to Harjot to explain the line of work performed in her lab and discuss the technical aspects of the ablation experiment.
Well, thank you, Anuradha. Can you do the stop share so that I can share my screen?
Yeah, sure.
Okay, so thank you, Pratima and Anuradha, for considering us to also have a part in this project. So as you have already seen that the neurons are needed for this kind of a behavior. So I just want to give a gist of this whole into that. This whole neuronal circuit has been developed by Sidney Brenner and John White long time back. That how a neuron is physically connected to each other. But it was very difficult to know that how functionally it is giving rise to the behavior. So one of the example I want to give is of Martin Shelby, who won the Nobel Prize in 2008. He used a simple touch circuit behavior. He wanted to know how the gentle touch circuit is being affected by the neurons. He used both the genetically and as well as the physiological approach for that. So as these C. elegans are transparent in nature, We can use the laser, which has been used in medical lines for cancer treatment to remove these neurons and see how a circuit is being made. So using these laser techniques, Martin Shelby and his colleagues could find out a set of circuit which was needed for the gentle touch circuit. This circuit has held up true up till now. So later on, other people have also found out the other circuits like chemosensation, thermosensation, etc. So previously they were using bright field to find out a neuron and ablate a neuron. But now we can genetically encode a neuron with the green fluorescent protein. So we can see a neuron. So we can precisely cut the part of a neuron with the laser. So this is an example where an axon of the PLM neuron has been cut. And this video is just showing that how this cut neuron was able to make growth cone formations, just like we see the growth cone formations in the peripheral nerve regeneration in humans also. So there was a great insight that we can get from this laser injury techniques. Our lab has also been using this laser injury techniques to find out how an axon, when it is injured by the laser, is able to regenerate back and make a regenerated synapse. Our lab could also demonstrate that this regenerative capability was reducing with the old age. And recently, some molecular mechanisms have been found out by our lab, such as LET7 microRNA pathway. It has also been shown in the retinal ganglion cell of the vertebrate rat retina that LET7 is playing inhibitory role in axon regeneration. Similarly, we could find out the insulin signaling and how physical exercise can enhance this regenerative potential. So this laser technique we have been using, and these are the questions that we were able to answer using this laser injury technique. So what is a laser and how does it work? So a laser, when it is used into the pulsed mode, it can make a very precise incision or precise scar onto a tissue. And as we move towards the infrared range, this laser can penetrate very deep into the tissue. And this infrared range is also having very less absorption towards the molecules such as water and HB. So we wanted this laser technique to work in our system. So we use IR. So IR, as it is infrared, this wavelength is not able to excite our fluorophores just such as green fluorescent protein. So we need 2 photons to excite these fluorophores to the excited state and give us the emission. So our lab uses this technique. So we have a system where we use 2 lasers. One laser we use for imaging the neuron, and the other laser we use for cutting the neuron. This is an example of exotomy, where you can see that as long as we can see the neuron, we are able to make a specific cut onto the axon of this neuron to create an axon break. So with this technique, we carried on to answering the question that have been asked in the Anuradha's and Pratima's project, that what will happen whether if we ablate this PDE and DVA neuron. So we used this strain with Anuradha only, and we tried to ablate the cell body of this PDE neuron. This is just an example to show that when we are able to make a cut onto the cell body of this PD neuron, we could see that the cytoplasm was scattering overall around into the surrounding, and there are some debris of this axon and the part of the cell body. This is the sign of the ablation. So we could do these ablations and further on could find out that how this DOP2 related functional behavior was working through this PD and DA neuron. So with this, I finish off our talk. I think we can carry on, Shona.
Well, thank you, Pratima, Anuradha, and Harjot, for beautifully explaining your work on dopamine signaling and also to establish C. elegans as a model to study these molecular players in locomotory functions. And also, importantly, for sharing this funny and serendipitous observation leading to the start of this project. So I think to our audience, if you have any questions, please feel free to write on the chat box, but I can carry forward the conversation ahead by asking a question to all the trainees first, and then I'll come to Anindya and Kavita. So to all the trainees, I think you guys have used a huge range of techniques to answer some of these research questions. How easy was it during your grad school or postdoctoral work to learn these techniques? This is mostly a question to our audience who are like undergraduates or masters now and maybe just starting their PhDs. So it will be useful for them.
So I can answer that.
Yes.
So first of all, that the technique that we first of all, the question is more important. And regarding that, if we have different type of techniques to answer a question. And one thing that we need to take it into account that like if we can know the technique, but troubleshooting is an everyday thing. So just learning the technique or just taking a course is not enough. We need to know, we need to be habituated with the system, with each and every small part of it, so that because one day like some moisture will come or some humidity will come and the laser will misalign. So if we will be able to troubleshoot it, we will able to make it work. So we cannot expect the ideal conditions and have just one part of training session and we will be good to go. So I think that troubleshooting is also a necessary part of it, and we are always in a learning process. Like now also, if I am going back to the system, I learn something new into it. So I think that is the part of it that we need to be always in a troubleshoot mode. Yeah.
Absolutely. Pratima, Anuradha, you want to add something to it?
Yeah, so actually, even when we are doing lots of these essays, so it is like it doesn't work in a single go. We have to, when we got this behavior, we had to like repeat it many times so that we know that, okay, this is what the behavior is for dope 2. And as Anuradha had discussed that initially we wanted to do the electrophysiology, but it was the lab in Australia that we were collaborating with. And so it was like they were not able to figure out how to do that behavior. So that was an advantage with Dr. Anindya's lab, since Anuradha could go there and she could perform the experiment herself, and then with the help of Harjot, she could ablate the neuron. So it's like, and when we are going for these collaborations, so it is very important, I think, that the lab we are collaborating with is nearby, so that in case they have any kind of issues, we can immediately solve the issue, because otherwise it is kind of a wastage of time at times.
Correct, absolutely. Yeah. Yes, Anuradha, you want to add?
Yeah, so yeah, it's all that what Pratima and Harjot have discussed, that troubleshooting, I think, is the main part. And we have different problems raising day to day in the same experiments that we are performing. Like someday we are going to see the behavior very beautifully, and the other day there is some or other change either in the like the temperature or the assay plates or the conditions. So even a very minute factor can affect your phenotype or the behavior. So yeah, so troubleshooting day to day with the experiments and the techniques is the most important thing we need to keep in our minds.
Absolutely. Okay, thank you, everyone. I think we've got a couple of questions from the chat box. I'll ask them. Ramya Shekhar asked, what is anterior touch habituation and how long does it take for the laser damaged neuron to regenerate?
Okay, so I would like to answer the anterior touch habituation. So here what we do, that we take this hair, it is like we have to take a eyelash hair. So with that, instead of like using our normal pick, we use that, and then we just keep touching the worm, the anterior head of the worm. And initially it tries to move out of that kind of, when we are giving that kind of touch. So, but finally it will get habituated. So that is that it is getting now prone to it and it is adapted. So that is the kind of habituation. So that we study that which kind of molecules might be important for that kind of behavior.
Okay.
I can answer the second part of it. So when we do the exotomy, we can see that the growth formation starts like from 3 to 5 hours after the laser shot. So we are also recently doing dendrotomy, where we are also seeing that like at around 6 hours, we could see the filopodial structure that have been forming to the proximal end, and like the regeneration process has started. Like physically, we can see the protrusions coming out. So it's like 3 to 5 hours. That is the time point where we can see the start of the regeneration. But it eventually goes to 24 hours where we can see the full regrowth, and then we can take it forward. Yeah.
Right. Okay. Thank you. And I think Bhagyashree also have a similar question. How long does a laser take to kill a neuron? And the other question is, have you used DOP2 inhibitors in your experiment instead of ablation?
So for ablation techniques, if we are giving the laser shot, so we can see that like at around like 4 to 5 minutes, we can see that the whole part of the neuron has been like deformed. But there is another thing that like some neurons which are having too many of processes, they need part of the cell body will be gone, but other parts are still remaining, and that take like a Wallerian degeneration for it to get cleared out. So it does take from like a 5 minute where the clearance is also there to like some hours, like around 6 to 7 hours, where you can see that the total clearance is there. So that is the other part.
Okay, so actually we didn't use the inhibitor since we wanted to silence a specific neuron, and inhibitor would have been more of it would have inhibited the whole dopaminergic system. Since it was a specific neuron, so we needed something which would go to just that neuron. So that's why we had to undertake this kind of experiment here. And more, so that is.
Right. Okay. Thank you. And I think now I'd request Kavita and Anindya to join our discussion. And I think I'll have a first question to them is, I think we've already understood what are the questions that seeded this conversation, this collaboration. But if you'd like to share, like, how do you choose a collaborator? Because time management is one of the most important thing, because you will have your own labs work, and on top of it, you'll be collaborating with someone with the other lab. So how do you choose a collaborator, and how do you do the time management based on your own labs research work as well?
Shall I answer this question, Kavita, or you want to add?
Any of you?
You go ahead, Anindya, and then I'll
I'm sure this answer will depends on vary across the board of the scientists, various scientists. The way I look at it is it's a good idea to work in a field and also know your colleagues in the field. And for example, Kavita, Sandhya, me in India, all are working in neuroscience area using worm, but our interest research questions are different. In that process, what we are doing is also generating reagents and tools which are sort of complement each other's interest. And since we know each other, it's easy for us to ask whether request and whether I can get these reagents. Often I call Kavita for help with reagents, and this is the conversation is ongoing, and it's in terms of understanding our research questions. So in that way, I would say that it's a good idea to know the colleagues in your field, and then it's easy for you to approach and ask for reagents, or and plan for a collaboration. Kavita?
Yeah, I agree with Anindya that especially the worm community in India is very small, and most of us would be happy to collaborate with one another. I have actually collaborated a lot with people in India and abroad, mostly from a very technical point of view. So I'm very open to collaboration if this is something that my lab does fairly routinely. Then I would have no problem. I'm very open to collaboration. And usually I go to labs that do certain experiments like electrophysiology or regeneration in Anindya's case. I've also collaborated with Sandhya Kaushika for looking at vesicle movement. So something that they do fairly routinely, I would ask them for a collaboration, which would make a lot of sense. I feel that that is the best kind of collaboration where you're not impinging too much, making the lab go out of their way to do different types of experiments that are not in their domain. They're working in their domain and it's one question. And both, either they do the experiment or a student from our lab goes there and does the experiment. All of that works quite nicely. And if the experiment is to do with C. elegans, I've collaborated with Thomas Pukadel's lab in IISc Pune to look at just injecting dyes into C. elegans and looking at the localization. So that's something that we can very easily do. In that case, I have no problem. But if the collaboration is out of our expertise, then I would have hesitation to collaborate.
Right. Okay. No, thank you. That's beautiful.
And regarding Shaunak, regarding time management, I want to add that when we have a research questions, that's part of our research. We have to manage the time. It's to do our research. We are every day managing our time. That's not a problem. It's integrated into the work. And when in some cases when somebody approach you for your expertise, then I think the time management issue comes more. And I am very lucky to have my students and postdocs. Various cases it happened. When I have requested them, and they never rejected me. That when something I want to do for other colleagues. So I would say that you need to have a research team where they are also working in a collaborative atmosphere, and they are ready to help others.
Absolutely. No, that's very beautifully said. I think generating the resources that will benefit the whole community, especially as you've mentioned that the C. elegans community is very small, yet in India, I think it's really useful for the whole community to grow. I'll put the next question to Pratima, Anuradha, Harjot, and any one of you can answer it. Is if you can share any of the difficulties that you've faced during this work.
Yeah, I am the right person to answer this question. Okay, so everything was kind of smooth going in Anindya's lab. Harjot helped me a lot, but I also ablated this GVA neuron. Okay, so the marker was not that prominent, and me and Harjot, we faced quite a lot of problems figuring out how to locate this particular neuron and ablate it. So yeah, that was one of the difficulties that we had faced during this study. And the another thing that was kind of difficult was relocating all my strains and C. elegans from bringing them from IISc Mohali to NBRC, change of environment and all. I got a lot of mails in my strains, and then again I had to manage and grow all my strains. So this was kind of little bit time taking, but yeah, it was all kind of when the experiment was successful.
I think Harjot can explain more why the first difficulty arose and how it's often happens. Yeah, Harjot, you want to explain?
Yeah, so we have a 2-photon laser system, and these lasers are very sensitive to the weather and humidity. So whenever there is some misalignment, we cannot see the marker very properly. And this marker was already in a red protein, so it was already a problem with seeing that how we can see this neuron. So what we did is that we actually had two channels. One is the GFP channel and red channel. So we used both of the channels to have the precise accumulation, and also we used autofluorescence other than the GFP. So autofluorescence also helped us to locate the cell body, and after the ablation also, we could see that in the other channel that this small red blob is going off. So I think sometimes we need to just have a click that like the other laser is working fine, other channels are working fine, so we can go into that band also and have like like precise understanding that okay, we can ablate this neuron this way. So yeah.
Right. I think, Arjun, I have a follow-up question. It's a very naive question. So when you do this laser ablation, is it a targeted approach, or which means it has no effect on the nearby cells?
So this 2-photon laser is a femtosecond laser. So as we are increasing the frequency of the laser, so previously people used microsecond laser. So microsecond laser could make more of the surrounding area scar. So as we are moving with the high frequency laser, this overall surrounding tissue damage is reduced. So in our case, ideally there should be no overall surrounding tissue scar. But because of some misalignments and you know that every day there is some change, there is some kind of a scar, but that doesn't hamper, that doesn't create any inhibitory role to the behavior. So I think we are good to go with that.
I see. Okay. So we have a question from Deepak Puria. Multidisciplinary approach like yours is absolutely necessary for efficiently address any biological question. However, every lab has their mandates either from funders or getting first author publications. During the process of the collaborative research, apart from the scientific discussion, when and how do you start the authorship conversation, and how do you reach to the end of authorship issue?
I could start off with this answer.
Okay.
So I've actually collaborated a lot. I have more papers which are collaborative papers than independent papers that are just our lab papers. And even my PhD and postdoc work, more than 50% of what I published has been collaborations. So I've been very lucky in that we've never had problems dealing with authorship. For some reason, I think everybody is aware of how much they have contributed to a manuscript and expect, that much. You expect the authorship placement as per what their contribution is. And I feel that if somebody's helped with one experiment, they obviously get authorship, but it usually is somebody else who's driving the project who gets the first authorship. And so a couple of papers we've had collaborations, which actually led to the collaborator doing a lot of experiments, doing around 35% of the experiments, which were electrophysiology. And that was a very important component of the paper. So in those cases, the author became a co-first author. But usually I found that this has never been an issue. And I've actually not started this discussion early on at all. I've started this discussion only after, only when we start writing the manuscript after the experiment is done. I don't know if that's a good way of doing it, but it's largely worked. So I don't know. Anindya, any thoughts from you?
Shall I add, Shwana?
Sure, absolutely.
Yeah. If you ask me, my answer is in a successful collaboration, less you worry about your relative position in the author list, is the better the collaboration. That's always worked for me. As Kavita also collaborated, I have been collaborating throughout my journey from PhD, postdoc, and while setting up the lab. I always asked for help, got the help, and help others. And in this collaboration, I think Kavita and me never talked about authorship. I think it was all done. So that is there. So in my opinion, it's a little bit, I say that it's a little bit of sense of sacrifice or sense of collegiality is required when somebody asks for your expertise and help. Why we want to collaborate is ultimately to help the project or the students, not to just for authorship. My job is to, or my team members' job is to ensure that thing is done. Sometimes we help others a group in Delhi area, talk to me or discussed with me, they want to set up in a C. elegans lab, we give strains and they get started. And, we don't even want authorship, we tell. All these things I think are helpful.
No, absolutely. I think he has a follow-up question on what about the lead authorship?
So I would usually go with the person who's leading the project, gets the lead authorship essentially. And if the collaboration is very extensive, then we would have a co-first authorship kind of arrangement. But usually it's, whoever's leading the project and, obviously thoughts are from that person, experiments are largely done by that person. That person would be the lead author.
No, absolutely. I think over the last 3 sessions of Crosstalk, we have a general consensus that everyone has agreed that it's a sense of collegiality and a friendship that goes in writing these papers and doing the work together, rather than discussing of this authorship before we even do the work. So I think we have, although our sample size is very low, we've been talking to more than 6 scientists.
I have seen my colleagues' experience that many of the collaboration died, at the middle of the project, while PIs become very possessive about their first authorship and so on and so forth.
I see.
So that I have seen. So ultimately, I think PIs should ensure that students or fellows who have done the work, their work is justified, their credits are justified.
No, absolutely. No, I agree. Do you have any tips for sustainable collaboration, mostly to young investigators who are just starting their lab? And I think you guys, both of you have collaborative work from your graduate school and postdoctoral work. But for example, someone who has never done this before and has just started their lab and would now like to collaborate, what will be your tips for a sustainable collaboration?
Anindya, you want to go first? Or...
I can keep on sharing, my experience. When I was graduate student, I did my PhD in TIFR. So by training, I was physicist and in master's biophysics specialization, but I choose, genetics. I decided to join a genetics lab so that I will learn biology. So, I was working on, understanding how cytoskeleton or motor proteins, help in the morphogenetic process during development. So with a particular focus on dynein motor. And when I was discussing my work in a meeting in NCBS, which was organized by Jitu Mayer. So there I met Richard Valley, who is like one of the discoverers of motor proteins like Ronville. So he came to my poster and I was discussing I had a model sort of with my genetics experiment, how these multiple subunits work together to activate the transport system. So then I approached him directly, whether, I can come to his lab and, whether he is willing to help me. I actually wanted to test that biochemically. And, scientists are, I think, quite generous. I have been very lucky. And Richard immediately invited me, during my PhD to come to Columbia University to do these experiments. And I have written a couple of grants, fellowship, all worked out. And in that process, I got my exposure to biochemistry and brought it to my lab, Krishanu's lab. Now I set up the biochemistry work with motor proteins. So it just, you have to make the move, I think. Talk to the right person, your peers, then it happens with a sincere interest. Kavita?
Yeah, I agree with that. I think you should talk to people, people who you would be interested in collaborating with. And I think you shouldn't feel too bad if they say they cannot do it also. I think it's both ways. Some people would be interested in collaborating, others for whatever reason might not be able to do it. And you should not just worry too much about it. I'm sure if you ask like 4 or 5 people for collaboration, 1 or 2 will definitely be interested in collaborating. But the one thing I would definitely suggest is go with what their expertise is. Do not just go randomly ask a worm lab, why don't you do these experiments, which they have no expertise in. So you should know your subject and you should be able to collaborate with people who would actually help, who work on what you're interested in showing in whatever system. So that is something that I would definitely suggest, that don't just go and say that can you do this experiment just because you're working on words. Come up with why it's required. So some of those things I'm quite, I think is very important, that, a little bit of the story, what they're looking at. Do not come to somebody initially and just say that, I want to do these experiments without any background. That becomes very difficult for, the person to also collaborate with you. So if you come up with something, if fairly well carved, the question fairly, well thought of, and some preliminary data that shows that you're going in the right direction, then most people would be interested in collaborating. I think we should just ask at that point. Yeah, but I think it's important that you don't just randomly go and say, this is what I'm interested in doing. Can I do it? Can I collaborate with you without some thought into the whole thing?
Right, absolutely. I think, Anindya, you have put a really interesting point on how your transition from biophysics to biochemistry and learning a new, like getting trained in a new discipline, bringing that to your own lab. So my last question to this panel today is, and maybe all of you can answer it in short, is your thoughts on the training and development opportunities that we get from this kind of collaboration and gaining a new knowledge that helps to initiate that in your own research. And is also a question to Anuradha, Pratima, and Harjot, how things that you've learned during your postdoc or grad school, a new technology or a new discipline that is helping your line of research now, and if you can share a couple of that. Maybe, Anindya, you can start because you've already said a little bit about your transition and how it has helped.
Yeah, that is sort of was an example how collaboration helped in learning and bringing and also initiating a new line of research in a new lab, sort of. And that has been sort of there for, I think, many scientists, that basically you don't know, you cannot know everything by yourself. Science is interdisciplinary, and collaboration has been inherent part of the research. And so in that process, I can keep on adding that when I went to Richard, after all these things, then also he suggested me to join a course which is conducted in Ujjwal Marine Biology Lab, and the famous physiology course at that time is run by Ronville and Tim Mitchison. And Richard actively pursued me, you should join this course. And it's a rigorous, 2 months course. I spent that 2005 summer just after finishing my PhD, and got exposed to various kind of scientists, got to hear interesting work, and I got the time to also decide what I want to do in future for my postdoc. So unless we are in a different environment, which is different from day-to-day work, we are not going to explore the new things.
Correct. No, I agree. Anyone else wants to add anything to it?
Okay, like you asked, how can we bring that training and expertise to our own lab or the institute in which we are working, like for a grad student? So I think the instrumentation facility is also very important, since I went there and learned the ablation process and experiment, got some training on it. I actually went twice, but since IISc Mohali and our lab didn't have this instrumentation and facility, so we could not proceed with that at IISc. So every time, like, we have to do like some ablation study or something, we will have to collaborate. Like after me, my senior Nagesh also went to Anindya's lab for same experiment, the same kind of experiment. So I think that instrumentation is also really important aspect. Like if you get exposure to it in your home institute, then you might be able to bring. That is kind of a drawback.
Absolutely. Okay, I think if no one wants to add anything, I think we are running out of time. I had a lot of questions in mind to ask, but I think in the interest of time, I have to stop for today. But thank you everyone, to all of our panelists, for taking the time and sharing your thoughts. I think we have all greatly benefited from these conversations. And thank you to our audience for asking these fantastic questions. Please stay tuned and follow Biologically Speaking on our social media platforms to get updates on our future session. We have a talk tomorrow to celebrate the World CRISPR Day by Dr. Debojyoti Chakraborty and Dr. Shobhik Maiti from the CSIR IGIB to talk about CRISPR technologies in India. With that, I'd request Dr. Mohit Jolly to share his concluding remarks. CROSSTALK has been his brainchild, and Biologically Speaking is proud to collaborate with him on this initiative. And thank you from my side. Over to you, Mohit.
Thank you, Shaunak. Can you hear me?
Yes.
Thank you so much, Harjot, Kavita, Pratima, and of course, Anindya and Kavita for accepting our invitation and for a wonderful discussion. I was taking some notes as you all were speaking, and I thoroughly enjoyed the candid and genuine conversation that we had. All the points that you mentioned about the worm community being so collegial, so collaborative, and of course, so interdisciplinary, where friends and colleagues help each other. That's very, very heartening to know. And I'm glad that will encourage many other students also to propagate that feeling later on, as well as they become part of the community in a more integrated way. I would also like to re-emphasize what Kavita and Anindya shared. So Kavita, your point about doing the homework first before randomly starting a collaboration, I think is excellent. And many people don't necessarily always do that. So that's I'm glad you pointed it out very clearly. And Anindya, thank you so much for sharing the journey that you had in graduate school. I'm sure that played a role in you being more open to collaboration, and of course, passing on that trait to the trainees in your lab as well. The aspects that you both mentioned about authorship never being an issue and also not discussed at the beginning of the project rather than at the end of it also speaks of sort of one of the highest levels of resonance, both in terms of professional and academic frameworks. So thank you so much once again. And I really enjoyed listening to all of you, learning from all of you, and wishing you all the very best in all your future collaborations. Thank you so much once again.
Thank you.
Yeah, thank you everyone, and have a great day.
Thank you so much. Thank you. We really enjoyed this.